Welcome to your last video for lesson 1. Today we're going to get into the physiological way that neurons are able to transmit messages. So we need to talk about steps of an action potential initiation and generation. So we've talked about what a neuron looks like at rest. At rest, a neuron has a lot of sodium ions on the outside of the axon and some potassium ions on the inside of the axon. This gives the neuron an overall negative charge. So what happens is we have to activate this neuron. So we have to have some sort of depolarization. And depolarization just means that we're now going to change the charge of the neuron. So what's going to happen is we're going to allow sodium to rush into your neuron. If we have all of these positives, okay, because remember sodium is charged, if we have all these positives rushing into the neuron, it changes what we call the polarity or the charge. So it depolarizes it. Remember, a neuron at rest is polarized. So a neuron that is now activated is going through depolarization. So first, let's take a look at these top two pictures, because this is what we've kind of talked about. So first, this is when we're at rest. And remember that a neuron at rest is polarized. And I'm just going to write P for polarized. This means that there is more potassium ions on the inside and more sodium ions on the outside. So it's negatively charged on the inside and positively charged on the outside. When we want to activate or depolarize a neuron, we have sodium rush into the axon here, okay, or the dendrites. And what happens is when we have all of these sodiums rush in, that means all of these positive ions are rushing into the cell. So what happens is the third picture. We actually depolarize it. So we have all of these sodium positive ions rushing in, and it's going to start to change the charge. It's all about the charge, guys. So it's now changing the neuron from being negatively charged to positively charged. Okay, step 2. So if the stimulus is strong enough, depolarization is going to really occur, and the polarity is going to be reversed, which now means that the inside of the neuron is going to be positive. Once we have enough of an electrical charge, we are going to actually cause an action potential to occur. An action potential is the start of the nerve impulse. Once you get a positive enough charge inside of the neuron, you're going to have an action potential, and there's no going back. Once you start to have your action potential, you're going to activate that neuron and have a nerve impulse. It's an all or nothing response. Okay, so that's that bottom picture again here. Once we have enough of a charge inside of the neuron, so now we're positively charged, we're going to create the action potential, which means that the nerve impulse has officially started. Now we have to move that charge down the neuron, because we just depolarized a small section. So propagation really just means that we're going to be moving the charge all the way down the neuron till it reaches the axon terminals. So you're going to depolarize one section of the neuron at a time. And really it just keeps going and going and going until it reaches those axon terminals. So we're looking at picture D now. So we have first depolarized this small section in the last picture. Now, when we depolarize this section here, it's going to cause this section to depolarize. Once that section depolarizes and enough charge has occurred, then it's going to depolarize this section of the neuron. And that's going to keep continuing until we reach those axon terminals, which is the end. So it's going to keep on going until it reaches the axon terminals, and that's called propagation. What happens when we're done? We have to repolarize. So basically, we have to repolarize if we want to have another impulse happen. So we start at the same spot that we first depolarized in. And all we do is we allow potassium ions to go out of the cell. So we're basically losing positive ions to the outside of the cell. Once again, this is going to cause the inside of the neuron to change back to positive. So now we're looking at E down here. So we're basically restoring that charge. So we're taking potassium ions, we're diffusing them out of the neuron, and those are positively charged. So if we're losing positively charged ions, the inside of the neuron is now going to be negatively charged again. And it moves in the same direction as our propagation did. So see how it moves in the same direction? So once this becomes positively charged, that's when we take this section here and we restore it to being negatively charged. Now wait a second. I just told you that potassium ions are diffusing out of the cell. But if you remember, when a cell is at rest, it has more sodium on the outside and potassium on the inside. We actually have to do one more step, and that's taking the potassium, putting it back into the neuron, and taking the sodium and putting it back to the outside of the neuron. And we do this by using the sodium-potassium pump. The sodium-potassium pump are found all along the neuron, and what they do is they take 3 sodiums, okay, so 3 sodiums here, and they take them from inside of the cell and they push them outside of the cell. At the same time, they take 2 potassiums and bring 2 potassiums back into the cell. Now you can do basic math. You're taking 3 positives and you're losing it, and you're gaining 2 positives. So overall, we have a loss of charge because 3 positives are leaving and only 2 are going in. So overall, eventually, you're going to have a negative charge on the outside, or a positive charge on the outside and a negative charge on the inside. So the events that we just talked about are for unmyelinated fibers. Okay, remember we talked about white and gray matter? So these are going to be for unmyelinated fibers. And this is really cool. This is where myelin sheath comes into play, and it's amazing. So we know that neurons that have myelin are much faster. But why? Well, it's something called saltatory conduction. And this is just fascinating to me. So here's your Schwann cell, okay, and there's your myelin all wrapped around your Schwann cell. And remember, when we looked at our depolarization, we had to depolarize every single section of the neuron. When a neuron is myelinated like this, we actually get to skip over the entire myelinated section. So it's so much faster because we're literally doing about 1/10 of the work. So if you look here, we've depolarized this section here. We've had sodium rush into the cell. And instead of having to depolarize this section and this section here, the myelin allows us to skip over that entire section and then just depolarize the small nodes of Ranvier in between them. So that's why when you have myelinated neurons, your transmission rate of a nerve impulse is so much faster because you're literally only having to depolarize about 1/10 of the actual axon. The last thing we're going to talk about is how we get that nerve impulse from one neuron to another. And this is actually, I think, pretty interesting. And if you took Anatomy 1, we kind of already talked about this when we looked at muscle contraction and how a neuron passes a nerve impulse and activates muscle contraction to occur. So what happens is the action potential and that depolarization is going to reach the axon terminal, which is our end. This is going to cause vesicles to be released from that neuron onto the next. So these vesicles fuse with the next neuron and actually release neurotransmitters into the synaptic cleft. Pretty interesting. So we release neurotransmitters into the synaptic cleft. This neurotransmitter actually goes across the synaptic cleft and binds to the next neuron. What this does is it allows sodium to rush into the next neuron. And what happens when sodium rushes into the cell? We're going to have an action potential. All right, last slide, reflexes. Reflexes are something that occurs in the CNS and the PNS. And really what I wanted you to kind of look at this is look at all the different sections here of a reflex. You have a receptor, which is going to be touch, pain, taste, smell. Your second thing is a sensory neuron. That's going to send information to the spinal cord or central nervous system. That's going to be your integration center. Once we integrate that information, we're going to use our fourth, which is the motor neuron. The motor neuron sends information to what we call an effector, and that's going to activate a muscle, like that's your muscle or your gland. So all of this together is what we call a reflex arc. So you have the receptor, which, like I said, this instance here is you stepped on a nail. So you stepped on a nail, that's your receptor. The sensory neuron is going to take that sensation of pain to your central nervous system, known as your integration center. The integration center is going to decide what to do with that information and then send the response through a motor neuron, which is going to activate the effector, which would be like a muscle in your leg that lifts your foot off of the nail. So all of these together are called a reflex arc. And just make sure that you know all 5 of these structures and how they kind of play in with a reflex. All right, so we made it through lesson 1. Nervous system is a very complex system. So stick with it. This is going to be definitely one of our longer lessons, so don't feel overwhelmed. And I really hope that you enjoyed the video, and please watch again if you need to review neuron physiology. It's not a super easy concept. It's not a super easy topic. So please, please, please watch again and just message your instructor if you have any questions.